We can hear you, yes.
Oh, great. So let me just.
And we can see the slide.
Let me just get the pointer. Okay. So may I start?
Yes, go ahead.
Okay. So good afternoon. My name is Bruno, and I'll talk a little bit about the light and rapidly regulated B-box proteins encoding solanolycopersicone genome. First, the B-box proteins, or BBX, are a family of zinc finger transcription factors that are known to have one or two B-box domains that are involved in protein-protein interactions, and they may have the CCT domain as well that also functions in protein-protein interactions and also has a canonical nuclear localization signal. If we look in Arabidopsis genome, we may find 32 proteins encoded by it, BBX proteins. They are divided in 5 structural groups that differ in relation to the number of BBX domains as well as the presence or absence of the CCT domain. From those 32 proteins, 23 up to date are functionally characterized. Of those, 14 are involved in the light signaling. Interestingly, 12 of these are from group 4 and 5. As a few examples, I can pinpoint the BBX28 that is degraded in darkness by the action of the COP1, but in light it interacts with the elongate hypocotyl5, HY5, and does not allows HY5 to bind in the promoter of the photomorphogenesis related genes. So it attenuates the transcriptional promotion of HY5. On the other hand, we also have BBX21 and as an example of a positive regulator of light signaling that not only increase HY5 expression, but it was recently described that it interacts with HY5 to promote the expression of photomorphogenic related genes. So what we know about the role of BBX proteins in light signaling came from Arabidopsis, especially in seedling photomorphogenesis. So little is known in other process as well as in other species. For instance, in tomato, we only have one functionally characterized BBX that it may be called 20 or BBX25 according to the authors. And we know that it's negatively regulated by the COOL4 DDB1-DET1 complex. And when overexpressed, it's clear that it promotes the accumulation of chlorophyll in fruits, as well carotenogenesis. As for the carotenogenesis, it is known that it promotes the expression of the phytoene synthase 1, that is a key enzyme for tomato fruit carotenogenesis. So our aim here was to provide a robust phylogeny to identify tomato BBX and to investigate via transcriptional profiling the group 4 and 5 encoded tomato encoding genes in the light signaling and along fruit development and ripening. So by searching tomato genome, we found 31 putative BBX protein, and when aligned with Arabidopsis, we found that the structure groups are all monophyletic. What was interesting is that this group here, that we call structure group 6, was previously described for Arabidopsis as part of the group 5. We could find a second BBox domain in the sequence here, and when we align the size of the protein is more similar to group 2 than to group 5. And if we look at the end of the alignment, we can still see some conserved residues of the CCT domain. So our interpretation of this is that these proteins here are indeed a subgroup of group 2 that lost the CCT domain rather than a new group or from group or similar to structure group 5 as previously described. Then using microtome cultivar, we profile in source and sink leaves. Here it's quite simple, the pattern we found with only two exceptions. The transcripts accumulate as the leaf becomes the source. Then we addressed in dark grown hypocotyls and cotyledons or the ones grow under photoperiodic conditions. And we found several patterns. Some BBX are not responsive in hypocotyls by our treatments, but they are in cotyledons. They may respond in the same way, increasing the transcription, the relative transcript amounts, or decreasing both in cotyledons and hypocotyls, or they may be responsive in opposite ways when compared hypocotyl and cotyledons. Only one was not responsive to this light treatment, while these other two here were responsive in hypocotyls but not in cotyledons. Then we addressed the long fruit development and ripening by sampling the immature green 3 and 5 stages, mature green, the breaker, 3 and 5 days of the breaker. As our cultivar has the wild type golden tulip to allele, we have the pedicelllar portion with fully developed chloroplast, the stellar portion with poorly developed chloroplast. So we sectioned the fruiting tree, discarded the transition portion here and analyzed these two here. First, as the pedicelllar and stellar portions displayed very similar patterns, I'm only going to show you the results from the pedicellar, and we find a wide variety of responses for each BBX. But what I'm going to show you more, I'm going to highlight is these three here, because they peak at immature green stages and decline afterwards. And also these three here, because they have a really clear ripening induction. And when we think about ripening induction in tomato, the first thing that comes to mind is the ripening inhibitor transcription factor. That's a master regulator of tomato ripening. So we searched in the promoter of those three genes and found putative ring binding site in all of them. And the binding of ring was confirmed by cheap qPCR. So we can see enriched regions in fruits that was transiently expressing the ring GFP fusion protein. So our conclusion is that the ripening-induced transcript accumulation of these three BBX may be a consequence of the ring binding and activating the expression as fruits ripens. Then, to further explore those six that I highlighted before, we made a new experiment using the wild type, the mutated uniform ripening that lacks functional golden tulip 2 GLK2 proteins, and two transgenic lines with fruit-specific silencing for phytochrome A or for phytochrome B2. What we know from these genotypes here from the literature is that GLK2 will activate chloroplast differentiation, phytochrome B2 can inhibit golden tulip 2, while phytochrome A will induce chloroplast division and biogenesis in a GLK2 independent way. Our results for the first three that peaks at mature green stages are quite simple. We see that in the absence of the GLK2 or FA, we have reductions at the green stages, while the absence of phytochrome B2 increases the transcript amounts. What this opposite behavior is similar to the behavior that we see towards the chloroplast differentiation. So what our hypothesis is that the proper chloroplast differentiation will lead to the proper induction of the accumulation of transcript of BBX 16 and 29. For BBX 28, we could not find any hints of regulation by the golden tulip 2 or phytochromes. So it's still unknown how it's regulated here. And for the three that are ripening induced, we got a really clear pattern. It's decreased in the three genotypes that we have analyzed. So for phytochromes, we hypothesize a direct effect over these three BBX here, while for golden tulip 2, it's a bit more tricky, the interpretation, because after the breaker stage, the expression as well as the protein GLK2 can hardly be detected. So it's hard to think that coding the direct effect of GLK2 that's causing these alterations here. So what our hypothesis is that the proper chloroplast differentiation at green stages will lead to a proper chromoplast differentiation at the ripe stages, and this proper transition will induce these three genes here.
Bruno, you have 2 minutes.
Okay, so to sum up, this part of our research that I have showed you, we were able to identify 31 putative BBX proteins encoded in tomato genome, and by the profiling of the genes encoding BBX from group 4 and 5, we saw different responses in the analyzed organs. So this BBX can be different organ specificities. And by analyzing the 6 BBX with the particular profile that we saw along fruit ripe envelopment, we were able to identify regulations by GLK2, phytochrome A and B2, and or RIN. So I'd like to thank all the authors, all the people that help us to gather and analyze all this data, our funding agencies, as well as our university, the organizers for the opportunity of speaking here, and you all for the attention. Thank you.
Thanks very much, Bruno.
Okay. Yeah.
Excellent talk.
Okay.
Right now, yes, we have a question for you. And the question is, are you planning to overexpress tomato BBX in different Arabidopsis mutant backgrounds?
First, we are looking at their specific functions in tomatoes. So we are working with generating lines to address their role in tomato physiology. So we are focusing first on this, but this is a possibility, but not for the near future.
Okay. And another question from Jose Angel Martin Rodriguez. He liked your talk, and a question about the phylogeny tree. Was that constructed based on the whole protein or just specific domains?
No, for this phylogeny, I use the whole protein.
Okay. Does the BBX protein slow the flow ripening? Not sure if I understand that one. Does the BBX proteins slow ripening?
Slow down the ripening process, maybe. So we don't know yet because we are first surveying and finding candidates to further studies. So now that we got those candidates, we are going to generate new lines and then try to check what are their effects over the vegetative growth and over the fruit physiology, as well as any other parameters like the content of nutraceutical components, for example.
Okay, thanks very much. And
Thank you.
I think I'll introduce the next one, Esther.
Sure, and one more thing. The speakers, please, if you can, turn on your video so we can actually see you.
Yeah, that would be nice. Okay.
I see Matthias.
Good. Matthias from Cold Spring Harbor Lab will present his talk now on the contribution of transposons to the structural variation and phenotypic diversity in tomato. So take it away.
Yes, hi everyone. Thank you, Christian, for the nice introduction. Can you see my slides okay?
Yep, everything's fine.
Great. So first of all, I would like to thank the organizing committee for putting together a great meeting. I'm really enjoying it so far, and for giving me the opportunity to present my research today. So I will talk today about the contribution of transposons to genomic variation and phenotypic diversity in tomato. So my talk will be separated in two main parts. So in the first half, I will be discussing the landscape of transposon-induced structural variation in the genomes of tomato. And then in the second part, I will focus on a particular transposon family that is a main contributor to the structural variation. So what do we mean by structural variation? So structural variation refers to large-scale structural differences within the genome that are inherited and polymorphic within species. So there are many ways to generate structural variation within genomes, and the most common being deletions and insertions. But today I will particularly focus on one type of structural variation, which are the structural variation induced by the activity of transposable elements. So transposable elements are mobile genetic elements that have the ability to replicate and jump around in the host genome. And because of these inherent features, they're actually a major source of structural variation in the genomes. So there are actually several examples of why transposable elements and their associated structural variants are important for the evolution of plant phenotype. And they've actually been instrumental in the process of crop domestication. The most maybe striking example is actually how the activity of a transposable element, which is called hopscotch, has, by inducing a cis regulatory mutation, altered the expression of the TB1 gene in maize and favorized the occurrence of an improved shoot architecture in order to give rise to the maize and this typical, apical dominance we know in modern maize today. So there are several other examples of how transposons have impacted crop phenotype, and most of the times in a very quantitative way. And it can be through loss of function by insertion into promoter of genes. In this case, the insertion in promoter of gene that regulates anthocyanin production in grapes, but also providing new regulatory sequences, such as this very nice example in blood oranges, where actually the insertion of a transposon in the promoter of the genes confers cold responsiveness to the anthocyanin producing gene, and then facilitates the accumulation of anthocyanin in blood oranges when they are exposed to cold temperatures. Of course, tomato is a fantastic model to look at transposon-induced structural variation and phenotypic diversification. As you will see here, several phenotypes of agronomical interest in tomato have been influenced by transposable elements. Fruit shape, inflorescence architecture, fruit color, leaf complexity, all those phenotypes are in some ways affected by the activity of transposable elements in tomato. But nevertheless, despite this huge importance of T activities in shaping the genome of tomato and facilitating the diversification of phenotypes, there's actually very little things known about really the contribution of Ts to structural variation at the population scale in tomato, right? So that's why it's very important to understand how much transposons are contributing to structural variation in tomato, to understand, how and when they trigger changes in gene activity, and why this change can actually be of interest for agricultural and breeding purposes. So to do that, I've taken advantage of the pan-SV tomato genome that we have been publishing earlier this year. So the pan-SV tomato genome aggregates over 200,000 structural variants identified in a very diverse panel of tomato accession that are covering the wide taxonomic diversity in tomato. Starting from the wild and species associated to Solanium folium, all the way to the most modern cultivars that we are using today in the tomato industry. So one main message from this pan-SV genome was two things. First, of course, when you compare modern tomatoes and their ancestral relatives, what you see that you have a huge increase in the number of structural variants you can detect in those genomes. But maybe, and that's even more important, what you see here is that insertion and deletion events that are here in blue and pink are actually making the large bulk of structural variants that are detected into the genomes. So what I wanted to know is how much of those events of insertion and deletion can actually be linked to the activity of transposable elements. So what we did is to extract the sequence that were associated with all those events of insertion and deletion. And we looked for the presence or a signature of T activity in those sequences. And what we found was very clear. When you take all the sequences associated to insertion and deletion in tomato, 80% of them are associated to transposon activity. That's huge. It means that, you know, over 150,000 of the structural variants detected in tomato are actually due to transposon activity. And even more than that, not all types of transposons are contributing similarly to the pool of structural variants in tomato. What I'm showing here is the relative contribution of different type of TE to the number of structural variants detected. And what you can see here is that the LTR retrotransposon, Gypsy, and Copia, here in purple and pink, are actually the major contributor in terms of structural variation in tomato genome. It really means that LTR retrotransposon are the main driver of genotypic genomic diversity in the tomato genomes. But even more than that, if you dig a little bit further and you actually look at the single transposon family level, what we see is very interesting. There is actually one transposon family, a single transposon family, that is actually explaining more than 23,000 of structural variants. And this family is called Rider. You can see it there. Rider, it's belonging to the Copia superfamily of LTR retrotransposon. And it's one of the most active transposon in the tomato genome. So why is Rider important? So we show it's creating genomic diversity. That's nice. But remember all those phenotypes I showed you earlier about, you know, the fruits and flowers phenotypes in tomato? All of those phenotypes are actually due to the transpositional activity of a Rider element. In every single case, Rider has triggered the change, the genomic change associated to those mutants, and then is a major contributor of phenotypic diversification in tomato. So because Rider is so important to genomic and phenotypic diversity, it was important to understand what are the molecular mechanisms conferring such an activity to this particular Rider family. And that will be the focus of the second part of my talk. So to understand how, you know, Rider behavior, so I went back to the Haynes reference genome, and the idea here was to, by de novo annotation, identifying all the intact Rider copies, meaning the copies that are potentially active in the tomato genome. So using a de novo transposon annotation tool, LTR-PRED, we identify 71 full-length Rider copies within the tomato genome. And these 71 copies can be actually split in two different subgroups. One group that is aggregating the evolutionary young copies, meaning the copies that are still potent for transposition. And on the other end, which is represent 2/3 of the Rider population in Haynes, Rider copies that have accumulated mutation in the regulatory encoding sequence and making them actually unable to transpose anymore. What is interesting to note is that most of the young Rider copies tend to be inserted in open chromatin region, close to genes, usually less than 2 kb away from genes, while more ancient copies tend to be located in heterochromatin and far from genes. So what explains the activity of the Rider transposon? So as any other gene, transposon expression is regulated by regulatory sequences that are located within their promoters. So in the case of LTR retrotransposons, the regulatory sequences are located in the long terminal repeats, LTR, that are flanking the codec sequence of the transposon. And when we look carefully at what's happening in Rider's LTR, we actually identified several cis regulatory elements and motifs that are associated with transcription factor binding. Interestingly, there is a significant enrichment in cis regulatory elements that are associated to dehydration responsive promoters. So based on this observation, we speculated that rider activity might be triggered by environmental stresses, in particular stress related to dehydration. So that's what we tested in vitro and in vivo. So I'm summarizing here our findings. So what we've been able to see is that there are several environmental. Okay, thanks, Esther. There are several environmental activators that will facilitate rider activity. So including drought, dehydration, soil stress, osmotic stress, and all those stresses are actually regulated through the production of abscisic acid. Tomato mutants that cannot produce abscisic acid are actually unable to respond to those stress, and rider activation is actually impaired in those ABA mutants. But of course, transposable elements are also strong mutagens, right? Because they jump everywhere in the genome, they are very likely to produce damage to genes and codec sequences. So you want to avoid that. So we also found that the genome is counteracting rider activity through an epigenetic response. And I'm summarizing here what we saw, and we've been able to see that in normal condition, there is a strong control of rider element through DNA methylation, and this DNA methylation being dependent of the production of small interfering RNAs triggered by the activity of the plant-specific RNA polymerases 4 and 5. So when in plants showing mutation for Pol4 and Pol5 induced through CRISPR, what we see is a reduction in small RNA targeting rider and a reduction in DNA methylation at specific rider copies that is associated with an increase in expression. And the last thing we wanted to know was, how widely distributed was rider in the Solanum genus. So what I'm showing you here is actually a distribution of rider elements within tomato relatives as well as potato and pepper. So this ridgeline plot shows you, so basically all the peaks you will see on the right-hand side of the plot are showing you intact rider copies, so very likely copies that are capable of transposition, while on the left-hand side are showing copies that are showing very limited homology to the reference rider, so very likely truncated rider elements or remnants of rider elements. So of course, as I showed you before, we see strong accumulation of intact rider copies in tomato, and we also see such a presence in Arcanum penilei and Abrochaitis. We don't see it in Pimpinifolium, and that's an interesting observation, and I'm happy to discuss that later, maybe in the breakout rooms. What is important here is that rider is not detected either in potato nor pepper, suggesting really that rider is Solanaceae specific transpose. So with this, so here are the two main take-home messages from my presentation. So transposons are the major source of structural variation in tomato genomes, and their activity have been really facilitating the phenotypic diversification during tomato domestication and breeding. And a major driver of this transposon-induced structural variance is rider. So rider, we have shown, is a very important and active transposon family in tomato, which is under environmental control and also epigenetic control by the plant host. So with this, I would like to conclude by thanking, so first the Liment Lab and especially Zach for his supervision. We have also a fantastic cohort of collaborators that were all very important for leading forward these projects. I would like to thank our funding bodies and thank you all for your attention. Happy to take questions.
Thank you, Matthias. There are two questions. One from Agustina Fortuny. Hi, Matthias. Is the PEN genome of the tomato available to download? I think that's our launch paper.
Yes, exactly. So that's the Alonge and Wong cell paper from earlier this year. So the data are all online. So all the sequences associated to the structural variation we code in this paper are available as supporting information. So yeah, they should be available. I'm happy to share the link for the paper if that's useful.
Another question, what percentage of the tomato genome is comprised of rider transposable?
So not that much. So it's really hard. So the difficulty here is really to, at some point, to identify what is rider and what is not rider anymore, because of the accumulation of mutation, truncation, and so on and so forth. But if you consider that, you have the 71 full-length intact rider elements. So each element is about 5 kb. So it's like, in the end, a very, very small fraction of the genome is made of rider elements. But of course, it will be, it will be a bit more because of the truncated elements and all the remnants that are harder to call.
And have you performed experiments to check the expression of rider upon drought or any other stress stimuli?
Yeah. So that's what we did in the paper. So we checked drought and dehydration, which were the main stresses activating rider. And we also tested other stresses like the cold stress, by example, because we identified cold responsive elements as well in rider LTRs. But surprisingly, rider wasn't affected by cold stress, by example. And it's not either activated by heat, which is also a common environmental stress that facilitates transposon activation.
Yeah, so there was a question also about, have you tried to let rider jump under adverse conditions? Yes, you have.
Yes, I have. And we see the transpositional intermediates. So when LTR to transposon are jumping, they first produce an extra chromosomal copy of themselves. And we have been able to detect those in the tomato genome, but so far we haven't been able to identify new insertions. So it really suggests that in addition to the environmental stress, you very likely need also probably a weakened epigenetic control. And unfortunately, we haven't been able to test that because applying stresses to epigenetic mutant in tomato is very hard, because the epigenetic mutants are usually not looking so great. So so that's definitely something we try to achieve.
There are more questions, but I think for the sake of time, we need to go to the last speaker. So please, if I didn't pick your question, come to the session after this, the talks.
Yeah, happy to take questions. Thank you.
Thanks, Matthias. Christian, I'll introduce Ashley.
Yeah, you do. It's the topic.
All right. So the last speaker of the talk is Ashley Snoofer from the University of Georgia. And she's going to talk about regulation of cell division and cell shape by tomato OFPs and trims to control early ovary development.
All right, thank you, Esther. You can see my screen and hear me fine?
Yes.
Perfect. All right, well, thank you first to the organizers for giving me an opportunity to discuss my work today. I'm very excited to tell you about my work on investigating the regulation of tomato fruit shape and the cellular mechanisms controlled by OFPs and trims to control this fruit shape. Well, if I can get my slides to advance. Hold on one second. Okay, so everyone is very familiar with the shape variation that you see in tomatoes in the supermarket, and the major genes involved in shape regulation in tomatoes are known, but the cellular mechanisms controlled by these genes is not known. So I'm going to focus in on the ovate family proteins. So ovate is the founding member of this protein family. There are 31 members in tomato. Natural mutations in ovate and another member of this ovate family protein family, OFP20, are known to regulate fruit shape. So here on the top, you can see ovate single mutants result in an elongation of the tomato fruit from that original small round shape. When you have a double mutation in ovate and OFP20, you end up with an actually pear-shaped fruit. So an elongated shape that's even further modified by the addition of OFP20. And something that we know that Esther's lab published a few years ago is that OFPs interact with another protein family called tone-1 recruitment motif proteins, or TRIMs. And here I'm showing you Solanum pimpinellifolium that has intergestions of these different gene mutations and a CRISPR mutation in TRIM5. And so you can see in the wild type, you have a round small fruit. When you have a double mutation in ovate and OFP20, just like in the fruits I was showing you before, you end up with a pear-shaped fruit. Ovate also slightly elongates the fruit shape. And then a triple mutant with TRIM5 actually partially rescues that elongated fruit shape phenotype that you see. And then something else that we know is that this phenotype is also visible within the ovaries. So even before fertilization and fruit development starts to occur, the ovaries are already elongated in that double mutant. And we see that partial rescue in the triple mutant as well. So this tells us that what's regulating this shape is actually occurring during ovary development rather than fruit development. And I just want to briefly mention that OFPs and TRIMs have been found to regulate organ shapes in a number of plant species, which is really exciting. So this OFP and TRIM interaction is likely more broadly important to understand for a number of plant species. So understanding the cellular mechanisms that regulate shape in tomato could have a broader impact on a number of plants. Okay, so just to kind of break down my question, in a really simplified view of organ morphogenesis to understand how ovate and OFP20 mutants result in this increased length in the proximal distal axis. I'm testing kind of 3 hypotheses here that I'll present to you today. So the first hypothesis is that to elongate the shape in the tomato, you're actually ending up with more elongated or larger cells. So the same number of cells in the wild type in the mutants, but they're elongated or larger. The second hypothesis I'm going to test is whether or not there's changes in cell division orientations. So again, same number or very similar number of cells within the organ, but there's a bias for cell divisions in a more elongating fashion in our mutants. And that's how you end up with an elongated organ. The final hypothesis I'm going to show you today is there's altered cell division frequencies. So in this case, there is a change in the cell number in the overall organ. And then perhaps if there's a reduced number of cells in the organ, then that results in a narrower organ that goes on to elongate more and looks more elongated in that axis versus the mediolateral axis. So those are kind of the 3 simplified hypotheses that are resulting in the morphogenesis of these different shapes. Okay, something I just want to touch on very briefly is that I'm showing you Solanum pimpinellifolium, and it's a really great developmental system because it initiates a new flower bud each day. So the developmental timing is very easy to track and to quantify. So here I'm showing you days post-initiation. So after that floral inflorescence is initiated. And so floral organs initiate sequentially. And so you'll start with the initiation of sepals here around 3 dpi, and then 4 dpi, the petals and the stamens will initiate. And then 5 dpi is when that carpel is specified, and 6 and 7 dpi are those really important stages when the carpel starts to elongate. And of course, the carpel will go on to form the ovary at a later stage. Okay, so first to show you when these mutants actually impact organ shape. So I showed you at anthesis, so that's a mature ovary. So we know that it's occurring at some point during ovary development. And here I'm showing you very early floral development. So this 4 dpi and then 6 and 7 dpi. And so at 4 dpi, this first bar is the wild type, and the second bar is the double mutant. We can already see a difference in organ shape in that remaining meristem that will go on to specify the carpel. So here I'm showing this in a little diagram up here in the purple region. So that's the remaining meristem, the region that we're measuring. So already the shape index, which is the length divided by the width, is slightly narrowed or a little bit more elongated. And I'll have this code on the side of each of those shape index graphs that I show you. And then TRIM5 in the triple mutant also impacts shape. So you can see here at 6 dpi, it's actually kind of flattening the shape a little bit. And I won't touch on this too much, but if you want to discuss it a little bit in the breakout section, I would be happy to. So basically, we know that these mutants are affecting remaining meristem shape very early in floral development. So it could be impacting the patterning of this organ somehow. Okay, and to go into that first hypothesis that I was talking about, so do these mutants affect cell shape or cell elongation? And so I'm showing you again here the mature ovaries. So we know that they do in fact affect cell shape. So here is the ovate mutant and then the double mutant. And they do have elongated cells. And I'm not showing the data here, but they also have slightly larger cells. So there is a change in cell shape and size. Again, TRIM5 does partially rescue that phenotype, and TRIM5 by itself also seems to impact cell shape in the opposite direction, where they're kind of flatter in the mature ovary. But again, these are mature ovaries, and a lot of things change as the ovary develops, and this could be an effect of mechanical stress or other factors. So we needed to look earlier into development to see if that initial change in remaining meristem shape that we see is actually a result of changes in cell elongation. So we in fact see that cell shape is not altered early in development. So again, here you have your wild type in the first bar, and then the double mutant in the second bar. And you see there's no significant difference in the shape of these cells. TRIM5 seems like it may have a little bit of effect earlier in development, but I won't touch on that too much here. But basically this suggests, at least in the ovate and OFP20 double mutant, that the driving factor is probably more of a cell division mechanism rather than a shape or elongation mechanism at this very early stage. So to go into that next hypothesis to test the cell division orientations and frequencies, I took the approach of EDU labeling. So as we're very familiar, growth is very heterogeneous in plants, both in time and space, and this can be seen at a tissue and a cellular scale. So it's really important to analyze this kind of phenotype over time to kind of get a trend through that heterogeneous growth. And EDU label allows me to do that. So I'm culturing floral buds for 9 hours with EDU. And the EDU labeling persists in the daughter nuclei. So I'm able to score cell division orientations by finding labeled daughter nuclei. So in the images I'm going to show you my EDU labeling, the EDU labeled cells are green, and then the blue cells is Hochsch, so a general nuclei marker that's marking all the nuclei. Okay, so to first describe the orientation of cell divisions, here I'm showing you wild type and ovate early ovaries, so the 2 to 4 DPI and then 5 to 7 DPI ovaries. And I'm scoring anticlinal, which would be kind of more horizontal cell divisions, and then periclinal, which will be more of those elongating cell divisions. And I made a little diagram down here to illustrate that as well. So in very early floral development, in that remaining meristem region, you see kind of equal amounts of periclinal and anticlinal cell divisions. And this makes sense developmentally. The organ is growing in both directions, so it would need both types of cell divisions. But there's no significant difference in the wild type and the ovate mutant. At 5 to 7 DPI, interestingly, we see a bias for those periclinal cell divisions. And again, as I'm illustrating on the right, this is when the carpel is starting to elongate. So that makes sense as well developmentally, that the organ is elongating, you would want more elongating cell divisions. But again, there was no significant difference between the wild type and the ovate mutant. So the orientation of cell divisions don't seem to be more biased in these mutants to make a more elongated organ. So it's probably not orientation of cell divisions. Okay, and then the last hypothesis I'm going to show you today is we tested the frequency of cell divisions. And here I'm showing you all the mutants. So wild type here is in blue, and then ovate is the orange, and TRIM5 in gray, and OP20 in the yellow, and then the double mutant is this lighter blue color, and then the triple mutant in green. So here I'm showing you very early, so 1 DPI, and then here's 2 to 4, 5 to 7, and then 8 to 10. And so in the ovate mutant by itself, we see a reduction in cell division frequency starting at 2 DPI through 7 DPI. The double mutant has an even further reduction in cell division frequencies, and you can see that already in the very, very early floral bud stage, and this persists through 7 DPI as well. At later stages, cell division is reduced in all of the mutants, so there's no really significant difference in the mutants at that stage. But these very early stages where the carpel is starting to initiate and just starting to elongate, there does seem to be a reduction in cell division frequencies. And I'm going to illustrate this here visually with some images. So here on the top, I have the wild types images. And so you can see lots of green labeled cells. And again, you can see a reduction in these green labeled cells at these stages in all three. But you can see very clearly in the ovate and then the double mutant here that there's far fewer green labeled cells. So there does seem to be a decrease in cell division frequencies in our mutants. Okay, so to summarize this, the organ shape changes can be detected very early in floral development, so at that 4 DPI stage. Cell shape is not altered early in development, but they are elongated in the mature ovaries, so there could be an interplay with both of those as the ovary develops that could impact shape throughout development. Cell division orientations are not altered in our mutants, but the cell division frequencies are. So we see that reduction in the elongated ovaries in the ovate and ovate SOP1, and again, very early in development. So kind of the thoughts going forward is that this is patterning a narrower carpal primordium, and this results as the carpal starts to elongate, a reduced width in the medial-lateral axis, and then eventually elongation in the posterior or the proximal distal axis. And I'll just kind of summarize this with a graphic. So here we have ovate and OFP20 mutants on the bottom. And so at those very early developmental stages, you actually have reduced cell division, but no developmental delay. So we know that these mutants develop on the same time course as wild type. It's just a reduction in cell division frequencies. And this reduction in cell division frequencies actually results in narrower carpal that then goes on to elongate just as it normally would. But because you have fewer cells in that medial-lateral axis, it results in an elongated posterior, proximal distal axis compared to wild type. So where we want to go in the future with this, so we want to do some further modeling of the changes in cell number and organ shape throughout development to really generate a good 3D model and understanding of how these different hypotheses could interplay with each other. And we need to identify the developmental stage where we actually see an increase in proximal distal length. So we've seen that stage where there's a reduction in the medial-lateral axis, but we need to identify when we can first see that elongation phenotype. And then how do OFPs and TRIMs alter cell division frequencies? So we know that OFPs and TRIMs can dynamically localize to microtubules. So we really want to investigate the microtubule dynamics and architectures within our different mutants in the developing ovary. Okay, and with that, I would like to thank firstly Esther as my PI, and then I would like to give a big shout out to Biao Zhang here. She did a lot of that work that I was showing in the published paper of Wu et al. 2018. And then Carmen, who did a lot of work on analyzing the early floral bud stages. And then of course, thank you to my entire lab, such a lovely group to work with, and my funding. And thank you all for your attention, and I'm happy to take any questions.
Thank you, Ashley. I think there's time just for one quick question.
Okay.
Really nice report from Alexander Goldsmith. Really nice report of pre-anthesis cell divisions. Have you looked at any post-anthesis cell division phenotypes associated with ovate and OFP20?
No, that's a good question. I haven't looked at that specifically. Based on expression, we think the majority of the phenotypes for ovate and OFP20 are probably earlier than anthesis, but there are other shape regulators that can affect fruit shape post-anthesis. And so I think looking at these kind of phenotypes in those mutants would be really beneficial, and it would be a very interesting study.
And maybe one more. Have you looked at cytokine and auxin content and signal transduction in these ovaries?
I have not personally looked at that, no, but there are some implications for changed hormone signaling. But I'm not quite sure how that would fit in with my results yet, but that's something really interesting to look at going into the future. So yeah, thank you for that question.
Well, thank you, Ashley, and thanks to all the speakers of this session. And I'm encouraging you to come out to the breakout session for more questions you might have, or just listen in on conversations. And there, it's in the chat, I believe, right? Yeah, so there's a link in the chat. It's also in your abstract book to go to the developmental meeting room that starts about right now.
Hey, thanks from my side too for everybody. Really nice talks, and talk to you in the breakout room.
See you, everyone.